Biology. This is a revision lesson on the topic enzymes, so grab your book and grab your pen and let us begin. The lesson objective is to explain how enzymes work as a biological catalyst. What are enzymes? That is the big question. Enzymes are what we call biological catalysts. When you hear the word biological, students, it is anything that is relating to living organisms. And a catalyst is a substance that increases the rate of chemical reactions without being used up or altered. So let's explain that a bit further. Back in the day, students, this is how I used to get my washing done. I would sit around a washpan and I would use my hands to do the washing. The washing would be done, the laundry would be nice and clean. The problem is those students is that it was so long. Now, this is how I get my washing done. I now have a washing machine and the washing is faster. And the good thing is that I can use this washing machine over and over, right? Because during the washing process, the washing machine is not altered. It's not used up in the process. And it's the same thing with enzymes. Remember we said that enzymes are catalysts. They speed up the rate of reaction just like the washing machine. And in the process, the enzyme is not altered and the enzymes are not used up. Just like the washing machine, it's not altered and it's not used up. So we can use the washing machine over and over again, just as our enzymes can be used over and over again. There is an initial minimum amount of energy required before any reaction can occur. This is called the activation energy. Enzymes work by lowering the activation energy. Let us look at this a bit further. I will be using this graph to show you the linkage between activation energy and enzymes. So if we're looking at the reaction where starch is broken down to sugar, students remember there is a minimum amount of energy required before this reaction can begin. And this is called the activation energy. This reaction is occurring without enzymes, and so the activation energy, students, is relatively high. And so if we were to add the enzyme amylase now to the reaction, this is what our new graph looks like. So the activation energy with enzymes present would be relatively low. And so this is important because enzymes allow reactions to take place at a lower temperature than normal. And this allows metabolic reactions now to occur rapidly in the human body at 37 degrees Celsius, which is relatively low and safe. Another thing that you need to know about enzymes is that they are protein molecules. And remember now, protein is made from amino acids. The chain of amino acid that enzymes are made from folds into a unique way so that each enzyme has an active site, right? So for example, that enzyme right there, the little grooves or the indentation would be the active site. That is where the substrate would bind. This is another example of an enzyme structure, and you notice how different this active site is from the first one. There is a theory that you need to know about, students, that is linked to enzymes, and it is called the lock and key theory. It is a simple model of how enzymes work. Each key has a particular shape that can only fit one lock, and so a car key would not be able to open the locks on your home door because the key and the lock is not complementary. And let us now look at how enzymes are linked to this lock and key theory. Similarly to the lock and key, a substrate will only fit the active site of one type of enzyme, and this is because their shapes are complementary. So let's look at this diagram. This is the enzyme, and remember the indentation represents the active site. Only one type of substrate can fit perfectly with this active site, and it's because the substrate has a shape that is complementary to the active site of the enzyme. Students, say for example, this is the enzyme protease and this is the substrate starch. Will there be a reaction between the two? Let us look at the active site. Do you notice that there is a gap between the substrate and the active site? It means that the substrate does not fit perfectly with the active site, and it's because the shapes are not complementary, so there will be no reaction. Remember, in order for a reaction to take place, the substrate must fit perfectly with the active site. Let's look at this one. So that's our enzyme, protease again, and that is our substrate. That must be protein because protein is broken down by protease, and the shapes are complementary, they fit perfectly, and so yes, a reaction would take place between these two. So let us now look at the full reaction. So we have our protease enzyme and we have our substrate, which is protein. When they react, students, these will be the product. So you'll have your protease and you will have your amino acids. Students, the protease enzyme is not affected by the reaction. It will not be altered, and so it can be reused over and over again to form amino acids when proteins are digested. Let us now do a little mini quiz to check your knowledge on the specific nature of enzymes. Question 1. Which type of enzyme breaks down starch? The answer is amylase. Question 2. Which type of enzyme breaks down protein? The answer is protease. Last question. Which type of enzyme breaks down lipids? The answer is lipase. So students, just a reminder, the enzymes are specific to the substrate that they break down. For example, starch will only be broken down by amylase. Why? Because the active site of amylase is only complementary to the shape of starch. Same thing for lipase. Lipase has an active site that is only complementary to lipids. Same thing for protease. The active site of protease is only complementary to the shape of proteins. I hope you understand this topic now, students. Thank you so much for watching and all the best in your exams.